Good afternoon, everyone. My name is Dr. Surbhi, Junior Resident in the Department of Psychiatry, and today I will be presenting my seminar on the topic of psychiatric aspects of gut microbiome.
We'll be covering this topic under these broad headings. First of all, a slight introduction to the topic, then scope, key definitions, core concepts, microbiota gut-brain axis, psychiatric implications, psychotropic implications, interventions, ongoing trials, emerging therapies, and later on followed by summarizing the whole presentation. Introduction. Psychomicrobiology investigates how alterations in the gut microbiome composition and function impacts the mental health and neuropsychiatric disorders, including depression, anxiety, bipolar disorders, and neurodegenerative disorders. Scope of the presentation. So study the bidirectional communication pathway between the gut microbiota and the brain, including the neural, immune, endocrine and metabolic routes. Examine microbial production of neuroactive substances such as neurotransmitters, short chain fatty acids and tryptophan metabolites that modulate the brain function. Investigates the effects of psychobiotics, which are beneficial live microorganisms and probiotics that can modulate the gut microbiota to improve psychiatric symptoms. Scope continued explores the role of bacterial extracellular vesicles as emerging mediators of gut-brain communication, applies the findings towards developing novel therapeutic strategies targeting the microbiome for psychiatric treatment and mental well-being. Key definitions that will be covered during the seminar: microbiota gut-brain axis. It is a complex bidirectional communication network linking the gastrointestinal tract, gut microbiota, enteric nervous system, immune system and central nervous system. It regulates physiological functions like digestion, immunity, neurotransmission and stress responses. Psychobiotics, a subclass of probiotics and prebiotics that positively influences the brain and the mental health through modulation of the MGBA. They exert anxiolytic and antidepressant effects via microbial metabolite productions and immune regulations. Core concepts. First is the colonization. So the establishment of the gut microbiota starting at birth, which is influenced by the mode of delivery, diet and environment. Healthy colonization is vital for MGBA development and mental health, which we'll be going through in the subsequent slides as well. Commensal gut bacteria. So the indigenous bacteria which resides symbiotically in the gut that performs the essential functions such as the metabolite production, the immune modulation and the barrier maintenance. Major phyla includes the Firmicutes and the Bacterotidetes. Neuroactive metabolites, so compounds which are produced by the gut microbiome such as the short chain fatty acids, the neurotransmitters including the serotonin and the GABA, tryptophan metabolites that influence the brain function and the behavior. Immune signaling, gut microbiota regulates the the balance between the pro and the anti-inflammatory cytokines which modulate the neuroinflammation implicated in the mental health disorders. Enteric nervous system, which is a subdivision of nervous system within the gut that coordinates gastrointestinal functions and communicates with the brain via the vagus nerve. HPA axis regulation, the gut microbiota modulate the hypothalamic pituitary adrenal axis, influencing stress, hormone release and systemic responses affecting mood and coordination. So the microbiome gut brain axis. Factors which affect the gut microbiome. So the connection between the gut microbiome and the central nervous system through bidirectional communication begins from the very intrauterine life, affected by many intrinsic and extrinsic factors which we will be dealing with in detail when we will be doing autism spectrum disorders. Factors which include the gut microbiome alterations. The most profound and the most studied factor is inflammation, acute and chronic stress. Vaginal and cesarean birth, lifestyle habits, living arrangements, both urban and rural, because they also somehow determine the parasitic infestations in the infants and in the adults. Dietary intake, medications intake, and the host's circadian clock. Neural pathway, the vagus nerve, a little bit about the functioning. So acts as a fast communication channel between the gut and the environment with central stress and emotional circuits. So the vagus nerve is most direct neural conduit between the gut and the brain. So it transmits the sensory information from the gastrointestinal tract to the brain. About 80 to 90% of the vagal fibers are afferent, meaning they carry information from the gut to the brain. Particularly targeted in the nucleus tractus, where it influences emotional and cognitive processing. Vagal stimulation reduces anxiety like behavior and depressive symptoms in animal models and humans. Enteric nervous system. So this is also known as the second brain. It almost contains approximately 100 million neurons, which are actually more than which are present in the spinal cord. And enteric nervous system dysfunction has been linked to the gastrointestinal comorbidities and psychiatric disorders. And one of the most profound ones is irritable bowel syndrome, which is seen in the patients with anxiety and depression. Irritable bowel syndrome forms the basic thin line between the two specialities, the gastroenterology and psychiatry. Hypothalamic pituitary adrenal axis. The HPA axis is the body's primary stress response system and plays a central role in gut-brain axis. And how does it do it? Because it has nothing to do with the gut. If you look at the whole axis, it has nothing to do with the gut, but still it has the most profound effect on the gut. So any stress activates the hypothalamus, which starts stimulating the CRH, which is the corticotropin releasing hormone. So now the CRH in turn triggers the ACTH production from the pituitary gland. So ACTH is responsible for the stimulation of secretion of cortisol from the adrenal glands. Cortisol is notorious for causing gut permeability, which is also known as the leaky gut, which we will be referring to in the further slides, and modulates microbiota composition and increases the pro-inflammatory cytokines, which can feed back to the brain and influence the mood. So stress-induced microbiota changes have been linked to increased anxiety and depressive symptoms. Immune system, the leaky gut. So immune system serves as a biochemical bridge between the gut and the brain. So increased gut permeability allows the bacterial lipopolysaccharides and metabolites to enter circulation, provoking systemic inflammation. Gut microbiota regulates the immune development and function. A healthy gut maintains the immune tolerance, whereas dysbiosis leads to immune activation. So this diagram also shows the same. So whenever there is some age-related dysbiosis or some ailment-related dysbiosis, so it causes the leaky gut. It impairs the permeability of the gut, which leads to all of these DNA damage, stem cell exhaustion, cellular senescence, all of this, which leads to aging and autophagy. Gut microbiota and microbial metabolites. So basically, this is the gut microbiota, which is predominantly present, and this is also known as, in a layman terminology, is the good bacteria of the gut. So the first class is Firmicutes, which has lactobacilli and the Clostridium, Bacterotidetes, which is bacteroids, and other bacteria like Acinetobacter, then we have Proteobacter and Fusobacter. Now the most important point, which is the microbial metabolites. So these gut bacteria, they are responsible for production of the microbial metabolites when they feed upon the food that is being fed to them by the human beings. And fiber, the dietary fiber, constitutes as the best food for these gut microbiomes for the production of microbial metabolites. So the first and the most important one out of them is the short chain fatty acids. So anywhere if there is decline in the short chain fatty acids, especially the butyrate, so it has been linked to mild cognitive impairment and Alzheimer's. Short chain fatty acids, they maintain the gut barrier and the blood-brain barrier, integrity, and they modulate the microglial activity and reduce neuroinflammation. And the best way for the production of short chain fatty acids in the gut is to feed the bacteria with fiber. Then comes the tryptophan metabolism. So it is altered altered tryptophan catabolites affect the serotonin levels, neuroimmune signaling and oxidative stress. Dysregulation of this pathway is linked to cognitive impairment and progression to neurodegenerative diseases. Trimethylamine N oxide, which is TMAO, it is elevated and it acts as a pro-inflammatory metabolite, and it is derived from the diet and the microbial metabolism, and is also associated with worsening of mitochondrial dysfunction, neuroinflammation and cognitive decline in the AD models. Then comes the secondary bile agents. These are the least studied ones, but have a very high potential for study and research in the future. So imbalances in the bile acid metabolism affects CNS immune responses and may either promote or protect against cognitive decline depending upon their profile and concentration, which is still under research as to which profile and as to which up to which concentration they are protective, and vice versa. Microbial dysbiosis in the mild cognitive impairment in Alzheimer's disease. Studies show distinct microbial profiles and metabolite patterns distinguish early cognitive impairment stages, suggesting that microbial metabolites could be both markers as well as contributors to the disease progression. Modulatory interventions, diets like Mediterranean diets in the mind that increase the short chain fatty acid production along with probiotics and lifestyle changes show promise in slowing cognitive decline by targeting the microbiome derived metabolites. Now, microbe and mother and child as a unit, microbiome and mother and child as a unit. So this is also very essential for us to understand that where do we get these bacteria from in the first place. So during the early development, the gut and the brain undergo parallel development during prenatal and early postnatal periods. Infants are born with immature vagus nerve and gut microbiota colonization begins at birth. So influenced by factors like delivery mode, breastfeeding, antibiotics and maternal microbiome. Maternal microbiome plays a very crucial role, which we will be studying in the subsequent slides. Microbiota role in neurodevelopment. So healthy gut microbiota contributes to normal brain development by producing neurotransmitters like GABA, dopamine, serotonin, which is modulating the immune responses and generating metabolites that influence neurogenesis, synaptogenesis and myelination. So germ-free animal models show impaired brain growth and behavioral deficits due to lack of microbial colonization. Mechanisms of communication. The axis function through neural pathway, endocrine and immune signaling, microbial metabolites crossing the blood-brain barrier and systemic metabolic signaling, microbial metabolites such as short-chain fatty acids and tryptophan derivatives regulate the brain function and behavior during infancy. Influences on cognitive and behavioral outcome, emerging infant microbiome patterns associated with cognitive functions such as language discrimination, social attention and neural rhythm tracking. Disruptions in early microbiota may lead to later neurodevelopmental disorders or behavioral issues. Nutrition and maternal factors. Maternal diet and infant nutrition significantly impact the microbiota development and thus the gut-brain axis. So breast milk provides microbes and metabolites essential for healthy gut colonization and brain development. Maternal factors. So modulating the maternal gut microbiome. So the maternal diet influences the composition of diversity of maternal gut microbiota, which in turn affects the microbe transfer to the fetus via the placenta and to infant during birth and breastfeeding. Bifidobacterium and lactobacilli while reducing pro-inflammatory and pathogenic species. Microbial metabolite production. A healthy maternal diet promotes the production of short-chain fatty acids by maternal gut bacteria. Immune modulation and protection. Maternal dietary changes shape the immune components of the breast milk, example the IgA antibodies, which support the infant gut colonization with beneficial microbes and augment immune defenses against infections. Impact on the neurodevelopmental risk, like we've already read. So unhealthy maternal diets like high in sugar, salt, saturated fats, alter the maternal and the fetal microbiome, leading to gut dysbiosis associated with increased risk of neurodevelopmental disorders such as autism. Supported evidences. Now, studies show that the infants of the mothers following a balanced diet, fiber-rich diet during gestation and lactation have healthier gut microbiota profiles, reduced infections in the early life, and better cognitive outcomes, emphasizing the critical window for prenatal and early life nutritional interventions. So, an overview of the gut microbiota affecting the psychiatric diseases. Now, this is something which is still under research. Not a lot of articles have been published, and it is still a topic for potential research. So, up until now, this is what has been inferred. So, in depression, there is suggested that there is reduced short chain fatty acid producers, so which increases the pro-inflammatory bacteria. So, therefore, there is neuroinflammation, which leads to impaired neurotransmission and thus the whole psychopathology of depression. In bipolar disorders also, a similar microbial change has been noticed, and it also leads to mood instability and HP axis dysregulation, just like we have read it before. Anxiety. In anxiety disorders, there is decreased diversity. So there is basically seen that almost all the flora undergoes decreased population, although the short chain fatty acid, altered short chain fatty acid producers. So heightened stress reactivity and activity behavior. Schizophrenia also, there is an increased lactic acid bacteria, so which alters the glutamate and GABA metabolism, and cognitive dysfunction and psychotic symptoms thus are the result of it. Autism spectrum disorder, wherein there is altered bacteroids and lactobacilli species, which causes behavioral and social deficits. Eating disorders, dysbiosis with pathogenic bacteria, which causes the gut-brain signaling disruption. Okay, so now this is the microbial taxa, which has been found to be predominant in these specific psychiatric illnesses, and starting with the Faecalibacterium. So Faecalibacterium, which when studied in the patients with MDD, bipolar and schizophrenia and anxiety disorders, was found to be consistently reduced. So therefore there is anti-inflammatory, this is actually an anti-inflammatory bacteria, so it produces butyrate, which is linked to better mental health, but it was found to be significantly reduced in the patients. Same goes for Coprococcus. This was also found to be significantly reduced, thus the butyrate production was found to be significantly reduced. Then coming to these three bacteria, the Egerthella, Lactobacilli and Enterococcus. So they were also studied in the patients of MDD, bipolar, schizophrenia, anxiety, MDD, bipolar, schizophrenia and MDD, bipolar respectively, wherein their abundance was found to be increased, and the abundance increased caused some pro-inflammatory, and these bacteria had pro-inflammatory potential. The same goes for streptococci also. Streptococci, the cultures were found to be enriched in the patients who were on psychotropic medications. So therefore, possible involvement in inflammation. Etopobium, it was also found enriched in the patients with mood disorders. Veillonella was found to be enriched in the patients in schizophrenia and MDD. And Escherichia and Shigella were found in bipolar, anxiety and schizophrenia patients, and there were increased pathogenic strains were found which were associated with inflammation. Bifidobacterium and bacteriotides, they are potentially beneficial probiotics, and they have shown their potential, their beneficial potential through these studies. Now talking about the gut microbiota in the psychotropic medications. This is also not widely studied, but researchers are still coming up. So patients who are on antipsychotics, especially the ones who are on antipsychotics for more than 2 weeks, there was found that there was decreased microbial diversity, especially the alpha diversity. So what was decided was so basically it was seen that the dose dependent reduction is present in the diversity. So as you increase the dose of antipsychotic, the diversity keeps on decreasing. So linked to metabolic side effects, reduces the short chain fatty acid synthesis. Antidepressants are actually said to have antimicrobial properties. So therefore, SSRIs are metabolized by the gut bacteria. Impact varies with drug and the patient microbiome profile. A lot of patients don't show any changes, but in a lot of patients, they come with very profound GI disturbances and GI symptoms because of the antimicrobial properties of antidepressants. Anxiolytics also, they are said that some benzodiazepines were affecting the microbial taxa, but studies are still not there are no impactful studies. Mood stabilizer and anticonvulsants are also notorious for altering microbiome diversity and metabolism in animal models. Opioid analgesics also induce dysbiosis with increased pathogens. So it is also linked to the gastrointestinal side effects and the behavioral changes. Now, basically what are the implications of the psychotropic medications in reference to the gut microbiota? First is the treatment response. So microbiome composition influences the drug metabolism and efficacy. So variable metabolism of SSRIs and the other psychotropics by gut bacteria may explain the differences in the patient response and drug bioavailability. Then coming to the side effects. Psychotropics alter the gut microbiota diversity and composition. So antipsychotics, they reduce the microbial diversity, potentially contributing to gastrointestinal side effects and systemic inflammation. Metabolic health. Microbiome changes due to psychotropic link to the metabolic dysfunction. Antipsychotics induced dysbiosis may worsen metabolic syndrome by reducing beneficial short chain fatty acid producing bacteria, leading to obesity and insulin resistance. Bidirectional microbiome drug interaction. Gut microbiome can also metabolize and modify the psychotropic drugs, influencing the pharmacokinetics and dynamics. Microbial enzymes, they transform drug, modulating their activity and toxicity, suggesting microbiome targeted therapies could optimize treatments. Personalized medicine. Microbiome profiling offers potential for tailoring psychotropic treatment and minimizing the side effects. So understanding individual microbiome drug interactions supports development of precision psychiatry and adjunct microbial therapies. Microbiome across the ages. So this is a pictorial representation of how the microbiome differs throughout the life of an individual. Like it was said, the microbiome population starts multiplying in utero itself. So when the fetus is growing, when the gut of the baby is growing, that is the time when the gut bacteria are also growing in it. So where does that baby get the gut bacteria from? So mother plays a very crucial and essential role. So there are 4 classes which have been widely studied: Firmicutes, bacteroides, Acinetobacter and Proteobacters. So if you see at the graphical representation at the top of the diagram, you can see that there is beta diversity and alpha diversity. I've mentioned it in the previous slide also that the alpha diversity is very important because these are the ones which are the good bacteria, which are responsible for good gut health, which are responsible for good physical health as well as mental health. So you can see as the child is growing towards the puberty and adulthood, the alpha diversity of microbiota is at its peak, and as the adult geriatric age sets in, the microbiota starts declining. Same goes for the beta one. Beta is quite profound during the in utero phase, but then as the child starts growing, it starts falling down, and with the geriatric age onset, it again starts growing. And immunity is directly linked to the alpha diversity of microbiota, which is the good bacteria. Now, so as you age, as a person ages, as healthy individual ages, so there is increase in the pro-inflammatory bacteria. So decrease in the beneficial microorganisms. So there is reduction in the beneficial bacteria such as Bifidobacterium, Ruminococcus and Prevotella with age. Changes in the microbiota diversities also happen due to our habits, due to the infections, due to our lifestyle, due to diet and due to stressors. Alteration in the core microbiota clusters also happens as we age. So there is higher levels of bacteroids and parabacteroids species compared to the cluster enriched in the younger population. Then impact of the external factors, accumulated effects of diet, antibiotics, lifestyle changes and physiological decline. And association with age-related diseases like Parkinson's, Alzheimer's disease or any metabolic disorders in the body. Microbiome and dementia. So psychiatry is seeing a transition wherein as far as the geriatric psychiatry is concerned, a lot of focus is being put on the psychomicrobiology aspect of the geriatric psychiatry medicine. So through this researches, this is what has come as an inference. So neuroinflammation. Dysbiosis increases the gut permeability and systemic inflammation, promoting neuroinflammatory processes in the brain. In Alzheimer's disease, this manifests as the microglial activation and higher pro-inflammatory cytokines level. Neurotransmitter alterations are also seen. So loss of beneficial microbes like lactobacilli and bifidobacterium reduces the key neurotransmitters like acetylcholine, GABA, serotonin, which are vital for memory, learning and synaptic functioning. Neuroplasticity and synaptic health. So microbial metabolites such as the short chain fatty acid and lactate enhance the hippocampal neuroplasticity, modulating the brain derived neurotropic factor and protecting against the synaptic dysfunction, which is seen in dementia. Direct effects on pathology. So Escherichia and Shigella abundance along with reduction in Eubacterium rectale are associated with increased amyloid beta deposition and inflammation in Alzheimer's brain. Autism spectrum disorder. So microbiota alterations are seen in the autism spectrum disorder. Children with ASD often present with gastrointestinal disturbances and microbial abnormalities. A lot of these patients do not have rhythmicity as far as the bladder bowel movements are concerned. Reduced abundance of Prevotella and Coprococcus and carbohydrate fermenting species has been reported. Gut permeability is quite significant and neuroactive metabolite altered short chain fatty acid levels may disrupt the brain signaling. Immune dysregulation, elevated inflammatory markers in ASD children correlate with gut dysbiosis. Now the use of proton pump inhibitors in the patients, which is quite seen amongst the population and is mostly iatrogenic. So they also have a very significant role on the microbial flora of the gut. So short term, if the patient has been using it from weeks to 2 months, so there is going to be initial alteration in the gut microbiota composition with decrease in beneficial short chain fatty acids producing bacteria and begin with minor changes in metabolite levels such as butyrate. And early dysbiosis effect starts, not only significant metabolite shifts yet. Then comes medium term, which is from 2 months to 6 months of use. So there is significant reduction in the short chain fatty acid levels, increases in lipopolysaccharide producing gram-negative bacteria and elevated systemic inflammatory markers start appearing. So dysbiosis has exacerbated. Then long-term use, which is more than 6 months to years. So there is sustained and pronounced decrease in short chain fatty acids, persistently high LPS levels during chronic low-grade inflammation. So there is going to be a long-lasting disruption in the gut microbial metabolite, correlate with systemic and possibly neuroinflammation. So decreases risk for metabolic and cognitive complications. So it is being studied, maybe this is the mechanism of action behind the depression which is being caused by the long-term use of proton pump inhibitors in the patients. Antibiotics. So the prolonged use of not even prolonged, excessive use of antibiotics causes alteration in the gut microbiota composition, which has been propagated by the WHO from many years. So the same goes here. So antibiotics reduce diversity and abundance of beneficial gut bacteria, increasing pathogenic species such as Escherichia coli and Clostridium inoculum. This significantly impacts the balance of microbial communities involved in the mental health regulation. Neurotransmitter disruptions, obviously antibiotic induced dysbiosis is going to reduce the brain levels of key neurotransmitters because there is going to be reduced production of short chain fatty acids. Then talking about the metabolic and functional pathways, so altered microbial metabolism, including changes in the pathway like lipoic acid metabolism, influences the brain function through production or depletion of neuroactive metabolites, impacting mood and brain behavior. Immune activation and inflammation. Dysbiosis increases the gut permeability, which allows the bacterial components such as the LPS to enter circulation, triggering a systemic inflammation. Vagal nerve and gut-brain signaling disruption. So antibiotics use modifies the vagal nerve activity and gut hormonal signaling, which is crucial for maintaining emotional and cognitive homeostasis. Behavioral manifestations. Animal models in some clinical studies show that antibiotic-induced dysbiosis provokes depressive and anxiety-like behavior, social deficits, and cognitive impairment. Now, what are the interventions that can be done? First and the most important one is the probiotics and the psychobiotics. So psychobiotics is a newly introduced class of medication, or you can say intervention, which has been specifically introduced for the gut-brain axis. So probiotics are live microorganisms that confer health benefits when consumed in adequate amount. Psychobiotics, on the other hand, they are the subclass of probiotics that are specifically exert beneficial effects on mental health through modulation of the gut-brain axis. So production of neurotransmitters like GABA and serotonin, it causes modulation of the HPA axis activity, enhancement of the gut-brain integrity, reducing the systemic inflammation, and reduction of the pro-inflammatory cytokines and increase in anti-inflammatory pathways. So what are the psychobiotics? So a lot of them have been studied, but these are the ones that have been most widely studied and are being used by the psychiatrists in the Western world. So strains like the Lactobacilli plantarum, Bifidobacterium adolescentes, and Akkermansia mucinellifera yield antidepressant, anxiolytic, and pro-cognitive effects in the preclinical and some human trials. Psychobiotic intervention can reduce serum corticosterone, increase anti-inflammatory cytokines, and enhance levels of serotonin or dopamine in the brain. Recent clinical reviews and meta-analysis highlights that psychobiotic, stress-reducing, mood-enhancing, and cognitive supporting effects with minimal side effects compared to synthetic psychotropics. Other interventions. Probiotics and prebiotics, so supplementing beneficial bacteria like lactobacilli and bifidobacterium on providing dietary fibers enhances growth of protective microbe, help maintain gut barrier function, and reduces pathogen colonization during drug treatments including antibiotics. So phage therapy and targeted microbial depletion. Selective removal of pathogenic or overgrown microbes through bacteriophages or antimicrobials aimed at specific bacteria help rebalance the microbiota without broad killing effects. Postbiotics, purified microbial metabolites or components that support gut ecosystem health without requiring live bacteria may mitigate drug-related microbiome disturbances, which is somewhere now become a part of guideline as well. As if a patient is being prescribed antibiotics, they are supposed to be prescribed postbiotics as well. Now this is the latest intervention, which is the fecal microbiota transplantation. So in the FMT involves the transfer of stool from a healthy donor into gastrointestinal tract of a patient to restore a balanced microbiome. The process involves screening a donor for infections, processing the stool into a liquid or capsule form, and then delivering it to the recipient's digestive tract via methods like colonoscopy, upper endoscopy, enema, or oral capsules. Now limitations. Lack of standardized protocols, concerns about the safety, long-term effects and donor selection, and ethical considerations in psychiatric use. Now this is the dietary intervention, which is the easiest one to be done, and which is something which has to be done by all in their day-to-day life. The most studied and the excelling out of all the diets is the Mediterranean diet, which is high in fiber and polyphenols, omega-3 fatty acids, fruits, vegetables and whole grains, promotes microbial diversity and anti-inflammatory microbial metabolites. The SMILE trials showed that dietary improvement with a modified Mediterranean diet significantly reduced depressive symptoms and compared to the social support control. Ketogenic diet, a high fat, low carbohydrate diet that modulates the body towards fat metabolism. According to several clinical studies, ketogenic diet showed a positive effect on patients with Alzheimer's disease through elucidation of levels of high blood ketones, which improved cognition and memory. But with ketogenic diet, high fiber has to be added because it is low in carbohydrate. So you have to add high fiber, otherwise it is of no use. Then fermented foods, a lot of Japanese diets are coming into the limelight these days because of the fermented foods that they consume. So that is also being studied. Ongoing trials. This is in a very naive stage of psychiatry, psychomicrobiology. So a lot of trials are being done, trials which are investigating the probiotics and the prebiotics in depression, anxiety, autism spectrum, in Parkinson's disease, aim to evaluate efficacy in symptom reduction and cognitive improvement. Fecal microbiota transplantation, which we just discussed, that is also under trial. And then which is being tried on patients with depressive disorders and neurodegenerative conditions. Studies are assessing the dietary interventions also, including more plant-based, high-fiber diets to support beneficial microbial metabolite production and enhance gut-brain signaling in psychiatric and metabolic disorders. This came after the increased colonic cancer sort of epidemic in the America, that they started focusing more on the plant-based diet. Emerging interest in microbiome-derived nanoparticles and engineered probiotics designed to target specific gut microbial communities or deliver neuroactive compounds in gaining traction as novel drug delivery methods. A lot of pharmaceutical companies and nutraceutical companies are focusing on this. Emerging therapies, first is metabolite centric therapies, like something which is going to be aiming at modulating the levels of short chain fatty acids, tryptophan metabolites and bile acids, targeting the intestinal barrier, blood-brain barrier and peripheral immune system. Integration of digital mental health platforms with microbiome targeted treatments is an emerging approach for personalized and synergistic care, particularly in adolescent depression. Personalized psychiatry. The future of gut-brain axis is based on interventions where in the interventions will be focused on the personalized psychiatry, where treatment is tailored based on individual microbiome composition. Microbiome sequencing, which enables identification of microbial signatures associated with psychiatric disorders, predictive biomarkers, wherein the specific microbial taxa may predict antidepressant responses, susceptibility to relapse. Precision interventions, probiotics, diets, or FMT can be targeted to correct individual microbial imbalances. This is a basic diet plan which everyone should follow. This is more like a Mediterranean diet plan, which can be included along with the Indian diet plan as well. High dietary fibers, plant-based foods, balanced macronutrients should be there, fermented foods. In North Indian diet, and even I think in the Indian diet, a lot of fermented foods in the form of pickles are also present. Then we have kanji, so that is also quite indigenous. Limited processed foods and added sugars, and adequate hydration. Hydration is essential for bowel movements also. These are my references. Thank you.